We are so excited to be welcoming everybody to this half-hour webinar session where we'll be talking about wild sourdough. As you can see here on our front page here, this project is made possible by the Museum of Life and Science and by NC State's Department of Applied Ecology, with whom we have some wonderful guests who are here to have a conversation with. And of course, the Wild Sourdough Project also does include a wonderful full list of other partners that are participating as well. And I will let maybe Lauren or Erin or Caiti drop a link to the project page into the chat box there in just a second. We only have half an hour together this morning, but we so appreciate you spending some time with us. Before we get started, I have just a couple of remarks before I turn it over to Peregrine and our lovely panel. And for those of you who are just joining us, feel free to introduce yourself there in the chat box. This is a webinar format, so you won't be able to see yourself, but you will always be able to see our full lovely panel that we have here today. Before we get started, welcome and thank you for joining our webinar today. Today's interesting topic is wild sourdough, and we're going to be talking about the fascinating world of the microbial garden living inside of a sourdough starter. We have a lot to talk about and not as much time as I'm sure we'd all like, so I'll be brief with our opening remarks and then open it up to our panelists. My name is Max Cawley, and I'm the Program Manager for Learning Philosophy and Evaluation at the Museum of Life and Science in Durham, North Carolina. I'd like to thank all of you and our esteemed and illustrious panelists for joining us today for this conversation. Our format today will be as follows. Following these opening remarks, I'm going to open it up to our wonderful Associate Program Manager of the lab, Peregrine Braschi, to conduct a conversation about wild sourdough with our wonderful panel today. If during this time you've got comments or questions or things like that, you can drop them into the chat box, though there's a few notes about that chat box that I'll note here in just a moment. Just a few things before we get underway. I want everyone to keep in mind that there are likely children present or watching in the background, as we're all figuring out how to live our family and work lives in the same space. To that end, we're maintaining a pretty strict code of behavioral conduct for this panel, and Zoom's functionality allows us to mute or remove people from the webinar if they're acting in a way that is inappropriate or inconsiderate for people watching at home with their children around. We don't want to, and frankly don't anticipate having to use that functionality, but this is a reminder that now is a time for compassion, consideration, and kindness to one another as we navigate these trying times together. We have, however, muted participants and will keep them muted and taken away screen share capability as well as a precaution. Yeah, so again, feel free to introduce yourself there into the chat box. And finally, we ask for and appreciate your patience and understanding as we're all learning as well about these interesting new times, about parenting, of course, about living our family and work lives sort of in a really similar space, about hosting webinars online. And to that end, and without further ado, I'm going to introduce our lab manager. Peregrine, take it away, my friend.
Hey friends, thank you so much for being here. As Max said, my name is Peregrine Bracci. I'm the Associate Program Manager for the lab. Our panel today is called Get to Know Sourdough. It's a Q&A session with scientists affiliated with the Rob Dunn Lab in the Department of Applied Ecology at NC State. We're collaborating with the Rob Dunn Lab on the Wild Sourdough project, and there will be more information on that on the Life and Science homepage. There's also a link in the chat. Please join me in welcoming our panelists, Dr. Erin McKenney, lecturer and academic coordinator in Applied Ecology at NC State University, Lauren Nichols, research technician and lab manager with the Rob Dunn Lab, and Caiti Lahue, lab technician with the Heil Lab in collaboration with the Rob Dunn Lab. Thank you all so much for being here with us today.
Yeah, pleasure to be here.
Thanks for having us.
Yes, thank you. Excited to get going.
I'm excited to have you guys here. So the name of the project is Wild Sourdough, but it's not just about wonderful bread. It's an experiment to study microbial ecology. Can you explain to us what microbial ecology is?
Yeah, so I'll start, and then Lauren and Caiti definitely fill in the gaps. I'd say for me, microbial ecology is the study of microorganisms, right, which are teeny tiny. You can only view them with microscopes, right? So, but they're all around us, all over the world. So they're covering your body right now. They're on every surface in your home. They're outside. We breathe them in. They are an integral part of our world, right? And it's the study not just of the microbes themselves, each different type of organism, right? For example, you have thousands of species living on and inside of your body. So it's not just the categorization and the naming of all of those different types. It's also the study of how they get along together and how they interact within their environment. So these microbes not only have different interactions, some are competing for the same foods, some are maybe producing antimicrobial agents like warfare to, prevent others from growing. But some are working together synergistically. One man's trash is another man's treasure. One microbe's poop is another microbe's food, right? And some of these products that these microbes and functions that these microbes are performing are incredibly helpful for us as well. So you may not think of microbes as much more than germs, but gosh, the vast majority of microbes in our world are at least, if they're not beneficial, then they're just along for the ride. So. Is that
I think Erin sums that up pretty well.
Yeah.
Cool. As I'm getting the second question ready, now is a good time for some folks to show off their sourdough starters. I have brought mine, which I've had for like 3 years. I am hoping to crowdsource a name for my sourdough starter. So if you've got ideas on what I should name this thing, I've thought about it for such a long time and have never quite gotten there, but feel free to drop that into the chat box as well. And Lauren, Erin, and Caiti, if y'all have brought yours or other things, feel free to show those off too. In the meantime, our next question is, how does making a sourdough starter help us learn about the microbes in our environment?
Yeah. I guess I could field that if you like. And then Erin and Caiti can jump in. So microbes, because they're so small, and they're so hard to see and they're everywhere, they can be really difficult to study because they're so incredibly complex. There's so many. I mean, just on your body, you probably have thousands of different kinds. Some of them we've never identified before. We don't know what they are. We don't know what they do. And so they can be incredibly difficult to study. And the beautiful thing about sourdough is that it's sort of a simplified system. In some ways, it's like your gut. Some of the same processes that are happening in that starter are some of the same processes that we see happen in other environments. But because of the nature, just some of the characteristics of a sourdough starter, it just happens that it simplifies the system down to something that we can actually study and replicate. And there's some reliability about how that environment and ecology works, that we can really break down this really complicated system into something that is actually studyable. And the beautiful thing is you don't need a fancy title degree and you don't need really fancy lab equipment. Right now we're locked out of the lab too, but we're trying to continue doing science. And this is actually science that we can do in our own homes with the tools we already have in our kitchen. So it's a really beautiful way for us all to sort of pare down these really complicated dynamics into a much simpler system that we can study. And from there, we learn just some simple how microbes relate to each other, how adding nutrients or taking away nutrients influences those dynamics, that we can then use these as sort of understanding some basics about the microbes that we can then apply to some of these more complicated systems.
Yeah. That's beautifully said. And I especially appreciate, as a gut person, right? I appreciate the idea that a sourdough starter is kind of like a gut in a jar, right? But whereas You put a variety of different foods into your gut every day, right? So I'll share some of my other lacto-fermented goodies, right? If you're putting tomatoes with garlic into your gut, or if you're putting kimchi, which is a mixture of cabbage and ginger and radish and all sorts of different spices, and even fish sauce if you choose to go that adventurous route, right? Or if it's sauerkraut, right, which may or may not have caraway seeds or other flavorings, right? That's a huge diversity of food types and of nutrients. So you can imagine for each of those different types of foods, microbes, different types of microbes would be more or less successful at digesting those foods, and they would require different functions or different digestive enzymes to break those foods apart. But with a sourdough, you're still digesting sugar and starch, but it's a very specific, a very narrow range of nutrients, because it's always going to be flour, right? So it's always going to be the ground seeds of grasses or of other plants, right? So we have a more narrow nutritional profile. So by controlling that specific nutritional source and water, right, which is again going to be a pretty simple input, I think that's how we are able to really simplify the inputs and the microbial system that arises.
Right, and just to add some fun facts to all these wonderful descriptions, the microorganisms are actually eating just like you or I would eat these foods. They break it down the same way. And in fact, a lot of the microbes inside your gut actually allow you to get the nutrients out of the food, because we ourselves lack all the specialized material and enzymes to actually break down the food we eat. So it really is a simplified version in a jar, because these might be the same things that are providing nutrients to you when you eat flour, water, or tomatoes and garlic, or kimchi.
I love thinking about sourdough starter as like a mini laboratory. It's so cool. We have some really cool questions in the chat that I think are similar to our next question. I'm going to go ahead and read our next question. Is what do the microbes do to flour and water to make starter, and how does that affect the bread? This is kind of right along the same lines as some questions in the chat, such as someone who's never created sourdough starter, where do you begin? From Maya. And Alexandra Sirota says, why do they call it sour? Perhaps these are all related.
Oh, those are great questions. So I'll start, maybe, I'm not sure which order of those questions came in, but I'll start with, if you've never started sourdough, never baked sourdough before, now is a great time to start. We're actually going to be guiding you through how to make a starter with this project. So it's great if you've never done it before. I actually am doing a video series now that we'll add to the instructions we have on our website. And so this is my very first day one, and I just posted the video on our Instagram feed. We'll post it on our website, and I'll lead you through each day of what you need to do to keep your starter going. But what's happening inside that jar? So what you're doing, you mentioned that it's a very simplified system. So you have sugars and you have starches, and you're adding water to it. And so the microbes are digesting those sugars and starches. And what you have is bacteria that are digesting those. And these are lactic acid producing bacteria that produce acidity, which is where the sourness comes from. And what that acidity also happens to do is it suppresses the growth of some of the molds and the other, the fungi you don't want in your starter. But it turns out that the yeast can survive that acidity. And so you end up with this system that's the bacteria producing this lactic acid and the yeast, which can survive the acid produced by the bacteria. But the yeast are digesting the same sugars and the same starches and producing carbon dioxide, which is what's creating that leavening, which creates the bubbles and then creates sort of the rise in the bread. Maybe Erin can add to that if I've missed anything.
No, I think you did a fantastic job. Yeah, I think So at that start, just to emphasize that everything in the entire world can digest sugar and starch, right? They're pretty easy nutrients to break down, and so that's easy energy to come by. So that means not only can the lactic acid bacteria break down those sugars and starches, and yeasts can do it, but also molds, right? And molds grow really fast, and molds are not things that we usually want to have in our food. So one of the amazing things about these acid producing bacteria, you can consider them to be environmental engineers, right? In the same way that beavers build dams to turn streams into ponds, and then you have an entire different community of organisms that can live in a pond than would have been able to colonize a stream, or might have been blocked from a stream entirely. When lactic acid bacteria break down those sugars and starches and start producing acid, it drops the pH below the threshold level that a lot of different microorganisms can survive, right? So things like mold are very quick to take over foods and other things, but they don't do well in an environment that's acidic. So when you have the lactic acid bacteria colonize your starter and drop the pH, right, to make it more acidic, that's when you have this massive turning point. They're engineering the ecosystem to disfavor molds and to actually favor specific yeasts that are acid tolerant. And those yeasts are the ones that are producing the carbon dioxide and other aromatic gases, right? This provides a nice segue to the person who asked, why do we call it sourdough, right? So you have all of this acid production by the bacteria. And if you think about acidic foods, right? Things like citrus fruits, things like vinegar, number one, you'll start to drool, right? And we'll come back to that in a second, because that's a really an exciting thing, not just kind of a wipe your face kind of thing, right? So when you think about these acidic foods, you start to drool because it's so sour, right? So those acids that the bacteria produce actually lend a sour flavor to this sourdough bread. It's not always pucker your mouth sour, right? A lot of sourdough breads are actually very mild. You might only notice a very slight tang. So it doesn't have to be a super in your face punch in the mouth sour. It can be a very mild sour, but those acids still being present are still going to favor yeasts over molds, right? But the other awesome thing about these acids, not only do they lend a flavor and prevent the growth of mold, so they're extending the shelf life of your bread, but those acids are actually used as an energy source in your body. So when you take a bite of sourdough bread, or if you think about these sour foods, you start to drool, right? You start to salivate. That is because your body recognizes that those foods are food. They are good for you. They are highly nutritious. They should be digested. And that salivary response is your body trying to start the digestive process just on thinking about those foods. That doesn't happen if you think about a saltine cracker, right? And that's, your body just doesn't quite recognize that as food.
That's really fascinating. Does that coincide with the domestication of yeast?
So I think that has something to do with our long history of making bread, right? We know from hieroglyphics that have been uncovered, from ancient Egypt, that bread has been baked for at least 10,000 years. And now there's new evidence that even before we were growing these grains in crops, there's now evidence that, hunter-gatherer communities of people may have actually been gathering seeds from the wild and letting those ferment. So we may have been baking bread before we were actually farming. So that's really exciting, right? That's a very deep history. Human cultures have organized around microbial cultures, right? So in all of that time, if we have this deep history of making fermented bread, sourdough bread, that would be a lot of time for us. It's almost like the microbes are domesticating us, right? We have unwittingly, for thousands of years before we knew that microbes existed, we've been, favoring the growth of specific microbes to make bread, to sustain ourselves. And in all of that time, of course, our bodies would adapt and start to evolve to think of and respond to bread as food.
That's fascinating. And there's a few things that I find really, really interesting about that. One being that like this specific particular moment in history is a really interesting time for sourdough, specifically because there's no commercial yeast available anywhere in any stores. And so being able to get yeast that you can turn into bread, one of the best ways to do that is to domesticate it yourself. And just for clarity's sake, like I have my sourdough starter here, which folks have been submitting names for, which is really excellent. And one of the reasons that I'm so interested in naming it right now is the specific question about like yeast being domesticated. So humans have obviously domesticated, plants like these house plants next to me, or animals like my cat that is now peacefully sleeping over there instead of playing with my cables, which is really awesome. Is it safe to say that our sourdough starter is almost like analogous to the way that we have domesticated plants and animals? Could I refer to my sourdough starter as a pet? That's my main question.
That's It's an interesting question. So when you buy the yeast you buy from a store, usually it's usually one species, Saccharomyces cerevisiae, and maybe one of 3 strains of it. But there are hundreds and hundreds of species of yeast from other genera that you don't ever see in bread baking, that you don't buy from a store. And so when we talk about domestication of yeast, usually we're just talking about that one kind of yeast, the Saccharomyces cerevisiae, which is what we've domesticated. But there are many other kinds of yeast existing in the world. We actually don't know what they're doing and where they even exist and what they're doing in the wild. And that's what we're calling this wild sourdough, is because it doesn't only exist because we have it in a jar. It's actually doing something else in the world. And only recently have people realized that during the winter, for example, we've known for a long time that you find yeast on grapevines, for example. And so when you make wine, originally it came from the yeast that was on the grapes. But what happens in the winter when those grapes are on the vine? No one even knew where to find the yeast. And it turns out recently they discovered it exists in the guts of wasps in the winter. And so Caiti has actually been doing this amazing work of going out and looking at different insects to see what this relationship is between the yeast and these insects, because there's just so much basic biology of we don't even know what these wild yeasts are doing. So I don't know if we can, I think I'm torn on whether you can say that maybe the yeast that's specifically in your jar has become your pet, but in some ways I think it's still wild because the rest of it, we don't know what it's doing. We have no control over it. And so I think that's where this idea of wild sourdough comes from.
Yeah. So basically what you've done is you've given it a happy little environment that has lots of easy food, easy access to nutrition and hydration and everything it could possibly need. So it's decided to stick around, essentially. It could get up and float away on the next breeze if it wanted, but you've just provided it with a very nice home. And so it's sort of like the start of the domestication process, where it's like, yeah, I'll hang around this human who is providing me with lots of nutrition and an easy life. So
it's like a feral cat that's come by for some milk and is trying to decide whether to stick around or not.
Well, and that gets to one of these really excellent questions in here is, are there ways that we can create this like ideal home for them to stick around? Because in my experience, in the couple of years that I've been doing it, I've gone through some starters that have failed, and I have not really understood why. But are there, one of the questions here is, is there a benefit to keeping it in glass instead of plastic? Is there an ideal temperature? Do we know of an ideal like feeding schedule? Like, any idea on how to speak to any of those questions?
So these are fantastic questions, and I think we should start by saying we don't know the formal answers. No, there has been no scientific research to date to really say, yes, this is the way to, favor these microbes, or glass is better than plastic. There is a lot of anecdotal evidence, and one of the amazing things about working with all of all of these established professional bakers and home bakers who have a lot of experience, and newcomers to the project. You all bring such diverse perspectives and so much excitement and enthusiasm, and you're all so observant, right? Each of us is one individual and we are each a product of our own backgrounds and experiences. So we all bring different skill sets and kind of biases to the table, right? So I tend to think about things from a perspective of animal science, nutrition, and guts. And then, as a foodie, right? Also thinking about, oh, wouldn't this flavor combination be fun? But that's a very specific, distinct perspective to have. And I think for each of you who joins the project and who's sharing your thoughts, you are strengthening the questions and the approaches that we can use. And you're really adding to it. You are equal partners in this scientific endeavor. So that's what has me really excited. Every time that I hear questions, I'm like, yes, we're wondering that too. And we have no idea. So help us find out.
Yeah.
Sometimes we have, some ideas, like we do know that glass is much less permeable, right? Glass, if you just stick it in your yard or in a landfill somewhere, it'll be around for, thousands of years, right? Glass doesn't tend to break down. Whereas plastic will break down over time, and plastic can be a little bit porous. So I would think, a microbe might figure out how to hide in the plastic or even maybe digest a plastic over time, right? So that it might be more interactive with your sourdough than a glass would. But we have no idea. We have no idea how plastic versus glass is going to affect the microbes.
Yeah. So I guess I also want to back up. I'm not sure we've actually described what our current experiment is. And so what we're asking people to do is to create your own wild sourdough starter. And so we'll guide you through the steps, sort of like a recipe, but everyone is going to capture different yeast and different bacteria. And so you sort of have to learn the personality of, there's kind of like a personality characteristics of which yeast and which bacteria you get. And so they might need different things. And the same routine might not work for all of them. Some of them might digest the starches and the sugars much quicker than others. And so you might need to adjust your feeding. And so there's gonna never be one blanket prescriptive way to keep a starter. And that's why pulling together, what we're asking people to do is at the end, once you've made a starter or it didn't work, to just tell us some quick characteristics of your starter. Like, how fast does it grow? How high does it get when it's grown to its fullest for 8 hours? What does it smell like? And then just to take a couple photos and send us that. And so we want to know about the ones that work, but also about the ones that don't. We hear anecdotally a lot of people say, oh, it didn't work, so I threw it out. But we have no consolidated data about is there anything that's similar about these that didn't work, and why didn't they work? I mean, it might be that, oh, I forgot to feed it on day 3. And it's consistently, if you see people forgot to feed it on day 3, but the people who forgot to feed it on day 5, it still survived. That tells us something about these ecosystems, and sort of, we can think about them as an ecologist who studied forests and insects. I think of it as a succession. So when you clear cut a forest and there's just like nutrients, what are the first plants that colonize? And usually they're the really weedy ones that grow really fast and can really use those resources. And then slowly, some slower growing, hardier trees start to dominate the forest. And so we can think about the microbes in the same way. Which are the microbes that colonize really fast? And at what point is your ecosystem really stable? So we're really excited about the starters that succeed and what those characteristics are and sort of their personalities even. But also, if you try it and it fails, please submit your data too, because that's equally exciting and something we don't hear about. I mean, in science in general, we don't hear about the failures enough, but there's so much data we can gather from it.
I really love that connection, and one of my very, very favorite things about this project is the fact that we are all scientists and we are all contributing really important data to this project. Unfortunately, this is all the time we have for this panel. I know there's so much more that we could talk about, but I want to thank you all so much for your time. Erin and Lauren and Caiti, thank you so much for being here, for your time, expertise. Thank you to all of our viewers. Oh, thank you so much to Erin for putting that link again in the chat, because you can learn more about the project on that site. And you can also watch the Life and Science homepage for more information about the Wild Sourdough Project. And be sure to join us for our first Lab at Home live session on April 15th, where we can get the Wild Sourdough experiment started all together. There's more information about that to come.
Thanks so much for having us.
Yeah, absolutely.
This was a lot of fun, my friends. Thanks so much.
And good luck, everybody, with your starters.
Thanks so much, everyone.
All right, I'm going to go ahead and pause the recording here.